Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the basics of antibiotic resistance, from key terminology you need to know in this field to main types of resistance mechanisms we observe. Now, the initial discovery of antibiotics changed the course of human history. From the 1940s through to the 1960s, this was often called the golden age of antibiotics. New drugs were almost discovered yearly, turning once lethal infections like pneumonia, sepsis and tuberculosis into treatable conditions. And for the first time, bacterial diseases that had shaped human mortality for centuries could be cured with a simple course of tablets or injections. Now, the impact was staggering as global morbidity and mortality rates from infectious diseases fell dramatically. Surgical outcomes improved, and life expectancy started to rise. And at this point, confidence in medical progress was so high that in 1969 the US Surgeon General Dr. William Stewart famously declared, "It's time to close the book on infectious diseases, declare the war on pestilence won, and shift national resources to such chronic conditions as cancer and heart disease." Now, it was a statement born of optimism, and yet it was profoundly premature, and one which we might consider to be the ultimate faux pas. You see, bacteria were already evolving mechanisms to evade every antibiotic we had discovered, and it was the famous Dr. Ian Malcolm who said it best: life finds a way. Bacteria were essentially developing and adapting to antibiotics faster than we could develop new drugs, leading to antibiotic resistance, which by definition is the ability of bacteria to survive and multiply in the presence of an antibiotic at concentrations that would normally inhibit susceptible strains. In other words, treatment that once worked no longer does. But not all resistance is the same. Some bacteria are naturally resistant because of their biology, and we call this intrinsic resistance. This means they're inherently able to withstand certain antibiotics due to their structural or functional characteristics, such as an impermeable outer membrane or the absence of the target's drug site. Other bacteria become resistant through genetic change, and this is known as acquired resistance. In these cases, bacteria gain the ability to resist antibiotics through mutation or by horizontal gene transfer, acquiring new genes that protect them from drugs that would previously have been effective. Now, unfortunately, we don't just get resistant strains in the clinical environment. We have the emergence of MDR, XDR, and PDR strains, which stand for multi-drug resistant, extensively drug resistant, and pan-drug resistant. Now, for each of these, strains are resistant to multiple categories, which basically means they've developed or acquired mechanisms that protect it against antibiotics acting on completely different cellular targets, which presents itself as a major clinical challenge. And so, multi-drug resistant strains show resistance to at least one drug in 3 or more antimicrobial categories. Now, they are often still treatable, but our options are becoming extremely limited. Extensively drug-resistant strains are resistant to at least one drug in all but 2 or fewer antimicrobial categories, meaning they remain susceptible to only 1 or 2 classes of antibiotics at best. And then we have our pan-drug resistant strains, which are resistant to all drugs in all clinically available antimicrobial categories, leaving no effective treatments remaining with reliance on untested experimental drugs. Now, hopefully from these definitions you can see that we get a natural progression as bacteria become increasingly more resistant and more difficult to treat. Now, this process of resistant arising is through several key mechanisms, and while the specific details vary, they all serve the same purpose: to protect the bacterial cell from the antibiotic's action. Now, some bacteria will do this by inactivating the antibiotic itself, often by producing enzymes that chemically modify or destroy it. Other bacteria alter the antibiotic's target site so that it can no longer bind effectively. Some bacteria will reduce the permeability of their cell envelope, preventing the antibiotic from getting inside the cell in the first place. Others will actively pump the drug out using efflux pumps so that it cannot accumulate within the cell. And finally, in some cases, bacteria will bypass the blocked pathway entirely, developing alternative metabolic routes that allow them to survive. Now, what we can see here are the 5 major strategies bacteria use to resist antibiotics. So let's look at some key examples. So our first general resistance mechanism is antibiotic inactivation. Essentially, this is where bacterial enzymes either break down or chemically alter the antibiotic, changing the antibiotic structure so that it can no longer recognize or bind to its intended target. And as a result, the antibiotic loses its biological activity and the bacteria remains unaffected. Now, this is actually one of the most widespread and clinically important forms of antibiotic resistance, especially against older classes of antibiotics. And so when we talk about inactivation, this can be via 2 different mechanisms: degradation or modification of the antibiotic. Now, a good example of antibiotic degradation is through the production of beta-lactamase enzymes. These target the beta-lactam ring of certain antibiotics like penicillin, which we can see here. Now, the beta-lactam ring is essential for its antimicrobial activity. However, beta-lactamase enzymes enzymatically degrade this ring so that it no longer binds to penicillin binding proteins, meaning it can't block transpeptidation reactions during cell wall synthesis, leading to the production of functional peptidoglycan. Now, our other example is where antibiotics are inactivated through chemical modifications, and a good example is the production of aminoglycoside acetyltransferase enzymes, which add acetyl groups to certain aminoglycoside antibiotics like gentamicin, altering its chemical structure and reducing its binding affinity for the bacterial ribosome, meaning it can't block protein synthesis and translation will continue as normal. And so in both of these examples, there is a complete loss of antibiotic function, effectively making the bacterial cells resistant to the antibiotics. Now, our second general resistance mechanism is target modification. Essentially, rather than modify the antibiotic, they modify the target site of the antibiotic. Now, these alterations or modifications are often due to spontaneous mutations or the acquisition of new genes, and they prevent the antibiotic from binding to their intended target. As a result, the targeted process remains unaffected and continues without interruption. And so when we talk about target modification, this can be via a number of different mechanisms, but the principles are the same. Now, a good example of target modification is the alteration of penicillin binding proteins in organisms such as methicillin-resistant Staphylococcus aureus or MRSA. Now, despite their name, penicillin binding proteins carry out transpeptidation reactions, forming cross-links between 2 strands of peptidoglycan, an essential step that increases structural integrity of the cell wall. Now, if we move on to a different cell, one that has been treated with methicillin, we can see that the methicillin binds to the transpeptidase domain of the penicillin binding protein using its beta-lactam ring, stopping transpeptidation activity and preventing cross-link formation. And this is where our modified target comes in. Some Staphylococcus aureus cells produce an alternative penicillin binding protein known as penicillin binding protein 2A, encoded by the MEK-A gene, which they most likely acquired by horizontal gene transfer. Now, this alternative enzyme has very low affinity for various beta-lactam antibiotics, including methicillin, due to modification of amino acids in the active site, preventing beta-lactam binding, while still allowing its transpeptidation activity, helping to form peptidoglycan crosslinks in the bacterial cell envelope and making the cell resistant to the antibiotic. Now, our third general resistance mechanism is a reduction in cell envelope permeability, which in basic terms is where bacteria limit the entry of antibiotics into the cell. Now, this commonly occurs in gram-negative bacteria as they fortify their outer membrane and limit what can pass through by altering their porins, stopping inhibitory concentrations from accumulating in the periplasm or the cytoplasm. And so when we talk about a reduction in permeability, this can be via two mechanisms: reduction or modification of the outer membrane porins. Now, a good example of decreased permeability is through the downregulation and loss of OPRD outer membrane porins in Pseudomonas aeruginosa. Under normal conditions, they allow low molecular weight molecules to diffuse into the cell, and unfortunately, this also includes antibiotics such as carbapenems, which enter the cell, accumulating until an inhibitory intracellular concentration is met. Now, the promoter and the gene encoding OPRD can undergo mutation, meaning functional OPRD porins are not produced, resulting in a marked reduction of their numbers. This means antibiotics, such as our carbapenems, have fewer routes of entry into the cell, and so the antibiotic is unable to build up to inhibitory concentrations. Now, our alternative mechanism to decrease permeability is where we have an alteration to the porin, and a good example of this is outer membrane porin F in Escherichia coli. Under normal conditions, they allow low molecular weight molecules to diffuse into the cell due to its wide channel, and unfortunately, once again, this also includes antibiotics such as beta-lactams, which enter the cell, accumulating until an inhibitory intracellular concentration is met. Now, when certain antibiotics are present, through a signal transduction cascade, Escherichia coli can produce an alternative porin, which has a much narrower channel, which significantly restricts the movement of antibiotics such as our beta-lactams, so they don't go into the cell, and therefore they are unable to build up to inhibitory concentrations. And so in both of these examples, antibiotics are unable to accumulate within the cell and reach that inhibitory concentration, effectively making the bacterial cells resistant to the antibiotics. Now, our fourth general resistance mechanism revolves around efflux pumps, which are essentially membrane-bound transport systems that actively export various molecules out of the cell, including antibiotics. Now, it's important to remember efflux pumps evolved for normal physiological roles, and therefore we have some that we might consider to be broad spectrum, working to export a number of different structurally diverse molecules out of the cell, which has been shown to contribute to multi-drug resistance, whilst others we might classify as narrow spectrum, only exporting very specific molecules with certain properties. Now, a good example of efflux-mediated antibiotic resistance is the Pseudomonas aeruginosa MEX-AB OPRM efflux pump system. Now, if we were to treat Pseudomonas aeruginosa with fluoroquinolones which target DNA gyrase in the cytoplasm, or with beta-lactam antibiotics that inhibit cell wall synthesis, these antibiotics will enter the cell, bind to their target protein, and essentially inhibit growth. And this is where the MEX-AB OPRM system comes in. This trio of proteins span the cell envelope and allow various molecules from the cytoplasm and periplasm to be exported out of these spaces. Now, this system is always expressed, and what it does is it actively pumps out these antibiotics when they're present, and it does this at a rate which is faster than the rate at which the antibiotics are allowed to enter the cell. And what this ultimately does is result in an intracellular antibiotic concentration that is too low for either antibiotic to exert its inhibitory effect, making the bacteria effectively resistant. And hopefully you can see that this resistance mechanism will work best when it's combined with our previous mechanisms where the cell limits how much antibiotic can come into the cell. Now, our fifth and final general resistance mechanism revolves around alternative pathways and links into a few of our previous resistance mechanisms. You see, when we treat bacteria with antibiotics, we hope they will bind to their target, inhibit that process, and by extension, the cell itself. However, sometimes the bacteria will have alternative biological pathways to complete this process. Now, this mechanism is almost always linked to one of the other resistance mechanisms we've talked about, and we can go back to one of our early examples to see this. The standard penicillin binding proteins in some Staphylococcus aureus species are easily inhibited through the binding of beta-lactam antibiotics, blocking that crucial transpeptidation activity that is essential for maintaining cell wall integrity. Now, as we saw earlier, some strains will encode a second penicillin binding protein known as penicillin binding protein 2A, which can resist the effects of various beta-lactam antibiotics. And so this provides an alternative unrestricted pathway achieving the same end result of transpeptidation and cross-link formation within our bacterial cell. And so those are the 5 key mechanisms by which bacteria develop resistance. Each of these mechanisms reduces the effectiveness of our antimicrobial therapies in a different way, but the consequences are the same: reduced susceptibility and increased occurrences of treatment failure. And so, if we are to slow the emergence and spread of these resistance mechanisms, we need to consider not only how bacteria resist antibiotics, but also how we use those antibiotics in clinical and community settings. And this leads us nicely on to antimicrobial stewardship, which is all about using antibiotics in a wise and safe manner to reduce patient morbidity and mortality, whilst also slowing the spread of antimicrobial resistance. Now, the principles of antimicrobial stewardship can be summarized by the 5 Ds: drug, dose, duration, de-escalation, and diagnostics. This essentially means we have to use the correct drug for the pathogen that is likely to be causing the disease, ensure that we use the correct dose so that there is a meaningful effect on the pathogen, use the drug for the correct duration and no longer than necessary, de-escalate where possible and switch to narrow-spectrum antibiotics once culture results are available, and ensure that we are using appropriate diagnostics to guide decision-making. Now, one of the key aspects about antimicrobial stewardship is that it isn't just common sense. It's an evidence-based, multidisciplinary strategy involving each stage of the process, from microbiologists to pharmacists, clinicians, and the infection control team. And so when these measures are followed correctly, it results in better patient safety, fewer side effects, lower costs, and reduced selection for antibiotic-resistant organisms, ensuring our antibiotics remain effective for future generations. And with that, we come to the end of this video. I hope you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.